Today we will be presenting on bioconcrete, also known as microbial concrete, which is an innovative type of concrete that can repair its own cracks. Bioconcrete contains specific bacteria that remain dormant inside the concrete structure. When cracks form and water enters these cracks, the bacteria become active and start producing calcium carbonate, which is the main component of limestone. This process is known as microbially induced calcium carbonate precipitation, or MICP. The calcium carbonate produced by the bacteria fills and seals the cracks, preventing further damage to the structure. As a result, bioconcrete can improve the durability, strength, and lifespan of concrete structure while reducing maintenance and repair costs. In simple terms, bioconcrete combines biology and construction technology to create a self-healing material that offers a more sustainable solution for modern infrastructure. Although concrete is known for its strength and durability, structural degradation is unavoidable over time. The process usually begins with the formation of microcracks caused by factors such as shrinkage, temperature change, and mechanical stress. These cracks may seem insignificant at first, but they can gradually grow larger. Once cracks are present, water, oxygen, and harmful substances can penetrate the concrete. This illustrates deterioration and weakens the structure. Over time, the steel reinforcement or rebar inside the concrete may start to corrode. As corrosion occurs, the steel expands, causing further cracking and spalling of the concrete surface. As a result, regular maintenance and repair become necessary, leading to increased cost and resource consumption. Therefore, technologies such as bioconcrete have been developed to address this issue by automatically repairing cracks at an early stage, helping to extend the service life of structures and reduce long-term maintenance requirements.
Sporosarsina pastori, a bacterium that plays a major role in microbial-induced calcium carbonate precipitation, or MICP, which is used for self-healing concrete and biocementation. One important characteristic of this bacterium is its extremophilic resilience. It can survive in highly alkaline conditions with a pH of around 12 to 13, which is similar to the environment found inside concrete. This makes it suitable for long-term application in construction materials. Another key feature is its electronegative cell surface. The bacterial cell wall carries a negative charge, which attracts positively charged calcium ions. These calcium ions accumulate on the cell surface and serve as nucleation sites where calcium carbonate crystals can begin to form. In addition, Sporosarsina pastori is known as a constitutive urease factory because it naturally produces large amount of the enzyme urease. This enzyme drives the biochemical reaction required for biomineralization. The continuous production of urease allows the bacterium to rapidly convert urea into carbonate ion, which are essential for calcium carbonate formation. Now I will explain the biochemical pathway of MICP, which is the process which bacteria produce calcium carbonate. The process occurs in two main steps. The first step is urea hydrolysis, which acts as the catalyst stage. The enzyme urea produced by Sporosarsina pastori breaks down urea in the presence of water, as shown in the equation. Urea reacts with water to produce ammonium ion and carbonate ion. This reaction also increases the local pH, creating favorable conditions for mineral formation. The second step is mineralization, which is the actual formation of calcium carbonate. The carbonate ion generated in the first step react with calcium ion available from the surrounding environment. When this ion combine, calcium carbonate is precipitated as a solid mineral. The bacterial cell surface acts as a nucleation site where the calcium carbonate crystals begin to grow. Over time, these crystals fill pores, cracks, and empty spaces within the concrete structure. This process improves the strength, durability, and self-healing capability of the material.
This slide will show the autonomic healing cycle, which explains how self-healing concrete repairs cracks using bacteria. In the first stage, a microcrack formed due to mechanical stress or temperature change in the concrete structure. In the second stage, water and oxygen from the environment enter the newly formed crack. These elements act as a trigger for the healing process. Next, dormant bacteria spores that are embedded in the concrete detect the presence of moisture and oxygen. As a result, the spores germinate and become active bacteria cells. In stage 4, the bacteria consume the available nutrient and carry out metabolic activity. This process leads to the formation of calcium carbonate through a mechanism known as MICP, or microbially induced calcium carbonate precipitation. Finally, the calcium carbonate crystal fill and seal the crack, restoring the integrity of the concrete. Once the repair is complete, the bacteria return to a dormant state until another crack appears. This slide focuses on encapsulation and survivability, which is an important aspect of self-healing concrete. One of the main challenges is that bacteria cannot survive when they are directly mixed into concrete. The mixing process creates higher shear force, and the concrete environment is highly alkaline, which is it can kill the most of bacteria. Therefore, protective carrier technology are needed to improve bacteria survival. As shown on the left, the bacteria are encapsulated inside a protective structure. The outer shell, which can be made from materials such as keramzite or polymer matrix, protect the biological payload. Inside, dormant bacterial spores are stored safely until they are active by the presence of water and oxygen. Several carrier technologies have been developed for this purpose. Expanded clay oxide keramzite provide good physical protection because of its porous structure. Hydrogels and polyurethane are able to retain large amount of water, helping bacteria activation when crack occur. Microcapsules offer a more uniform distribution within the concrete matrix and can effectively support crack healing. This slide also highlights a modern advancement called lyophilization or freeze drying. This technique converts active bacteria culture into stable dry formulation, making them easier to store, transport, and use on the large commercial scales.
Next, in a strategic deployment of MICP can be applied in several infrastructure environments. The first is marine and saline environment, such as seawalls and coastal piers. These structures are highly affected by chloride exposure, which can accelerate rebar corrosion. MICP helps by forming calcium carbonate layers that protect and strengthen the materials. The second application is underground infrastructure, including deep tunnels, subways, and foundation. Since water leakage is a common issue, MICP provides an in-situ repair method without requiring major excavation or reconstruction. The third context is bridge and high-stress spans. This structure experience continuous stress and micro-cracking due to heavy loads and environmental conditions. MICP can seal cracks and improve the durability of the structure. Lastly, MICP is useful for geotechnical ground improvement. It can strengthen low soil, reduce soil liquefaction risk, and increase load-bearing capacity without relying on the toxic chemical growth. Overall, this application shows that MICP has great potential in creating stronger and more sustainable infrastructure. Moving to the performance outcomes and future development of MICP, the results show significant improvement in concrete repair and strengthening. Based on empirical performance data, MICP achieved a 70% reduction in permeability compared to untreated cracked concrete. This means the treated concrete become more water resistant and has improved durability. Besides that, MICP also show 30.13% strength recovery after healing. This indicates that the process does not only seal cracks on the surface, but it also helps restore the internal mechanical strength of the concrete. For future development, synthetic biology can further enhance MICP performance. One approach is stellar morphology, where genetic engineering allows better control over the rate and formation of calcium carbonate precipitation. Another approach is mechanical turning, where engineered microorganisms can produce larger and stronger calcite crystals. This allows researchers to improve the mechanical properties of concrete repair materials. Overall, MICP has strong potential as a sustainable technology for creating safe healing and more durable infrastructure.